Let's now look at glycolysis specifically. And we're going to flag up slightly different terminology that different exam boards have here. The process, of course, is the same throughout. So glycolysis involves splitting. So we start off with a 6-carbon glucose molecule. Briefly, glucose, as you all know from your carbohydrates chapter, is not linear like that. It's a circular molecule. So before that, the circular molecule has become linear. You don't need to know the enzyme involved in that or anything. So what are the 4 stages of glycolysis then? So we've got stage number 1, phosphorylation of glucose. I'm going to try and use slightly different colors here. You can do that on your end if you want. So first stage is the phosphorylation of glucose. So it's basically this step here. You can see 2 phosphates have been added to glucose. And that utilizes 2 ATP molecules. So the phosphate here from ATP, 1 each, goes to phosphorylating glucose. The next step is to break down glucose, splitting of the phosphorylated glucose here. So this 6-carbon molecule became a 3-carbon molecule. And this molecule here is called, so here's where it differs a little bit between exam boards. So it can be called triose phosphate, TP. This will be particularly for Edexcel B, but it can also be called glycerate 3-phosphate, glycerate phosphate in OCR. Or actually in Edexcel A, it's also called an intermediate molecule, whereas in Edexcel B, it's called glycerate 3-phosphate. You'll know, depending which exam board you are. And then finally, we've got the production of ATP and pyruvate. I'm just going to change color here. So production of ATP and pyruvate. So here's my production of ATP, which basically dephosphorylates, right? It dephosphorylates triose phosphate. So that phosphate can be loaded onto ADP and converted to ATP. So here we have our pyruvate. This is my pyruvate. And here is my ATP produced. What are the final products of glycolysis? And this is quite a common thing that you might be asked. So first of all, we've got the, I'm going to try and use different colors here as well for you guys. So first of all, we have the 2-NADH, so otherwise known as the reduced form of NAD. We then have also 4 ATP molecules that are produced here. So 4 ATP molecules that are produced from converting the triose phosphate or glyceryl phosphate into pyruvates, right? However, because we also have previously 2 ATPs used in order to phosphorylate my glucose, my net product in terms of ATP are 2 ATPs. And from this reduction step here, I'm just going to use a different color. From this reduction step here, I should have done this in green, guys, sorry, is my 2 reduced NADH. And it's these that are actually going to go. The ATP produced is just energy produced, right? And in anaerobic respiration, as we're going to see, this is pretty much all the energy that's produced. But in aerobic respiration, for the next steps, NADH is the crucial molecule that is going to facilitate anaerobic oxidative phosphorylation, as we're going to see. Next stages. particularly oxidative phosphorylation. Let's see an example question about glycolysis, which is actually an extended response question. Glycolysis is the first stage in both aerobic and anaerobic respiration. Outline the processes of glycolysis. 1 mark will be awarded for quality of written communication. This is also sometimes abbreviated as Q, like that. So outline. Let's break down our question. What is my context? My context is that I'm looking at the process of glycolysis. What is my command word? Like clarity of communication, of course. And to describe the main points, my command word is the outline. And finally, what is my direction? It's that the process, my direction is that I am only focusing on glycolysis, and I'm only focusing on the process. Sorry, what did I do? I'm only focusing on the process and not any kind of, what is its impact, it's important or anything like that. Take a couple of minutes to see if you can structure this answer. So just write out little bullet points of like what would be your 5 marks, basically. Don't write out full sentences. We don't have time for that. All right. Let's look at the answer together. So really, glycolysis really is, comes in 4 main points, right? And so we got 4 marks here, and then we've got the additional mark for the qualitative communication. So glucose is phosphorylated using phosphate groups from 2 ATP molecules. That's my first mark. Molecules are then transferred. 2 ATP molecules are transferred to the glucose. Sorry, guys. I didn't read the whole sentence properly. So this is my first mark. I'm explaining the phosphorylation. Note the amount of detail that I'm giving, right? I'm not just saying glucose is phosphorylated. Where are the phosphates coming from? And where are they going? From ATP to glucose. Forming and naming the molecule resulting, glucose phosphate. Glucose phosphate then split into 2 molecules of triose phosphate being oxidized before being oxidized to pyruvate. So splits into 2 molecules of triose phosphate. That's my second mark. Before being oxidized to pyruvate is my 3rd mark. The 4th mark is for stating the net production of ATP molecules. So net production of ATP molecules 2 and 2 molecules of reduced NAD are mine. And then I get 1 additional mark for quality of written communication.